Let the recording start. Good. So, welcome everybody for this evening session. Today we'll be quickly brushing about phenylalanine and tyrosine metab. Okay. Now, phenylalanine and alanine, the only difference is the phenyl group. This is alanine and this is phenylalanine. The only difference being that a phenyl group is attached to the alanine molecule. So, both of them to some extent behave similarly. Now, phenylalanine gives rise to tyrosine. Tyrosine consists of an OH group, which is not present in the phenylalanine. When phenylalanine gets hydroxylated, it forms tyrosine, right? So what is the structure of phenylalanine? What is the structure of tyrosine? Nobody is going to ask you this in the examination, but generally we should know what we're talking about. Alanine, as we all know, gets transaminated to form pyruvate. And we read in the carbohydrate metabolism that pyruvate can form lactate and pyruvate can form acetate. Phenylalanine also follows a similar process. Phenylalanine gets converted to phenyl pyruvate, phenyl lactate, phenyl acetate. So we need to understand this because understanding phenylketonuria, this concept will be very important. Right. Now, the most important step that all of you should know about is the conversion of phenylalanine to tyrosine. Now, phenylalanine, there's an enzyme called phenylalanine hydroxylase, which adds the OH group. We said that the difference between phenylalanine and tyrosine is the OH group. Phenylalanine hydroxylase adds the OH group. In order to do so, it requires the help of tetrahydrobiopterin. Okay? Tetrahydrobiopterin gets reduced. Tetrahydrobiopterin gets reduced to dihydrobiopterin. Now, after the cycle is over, dihydrobiopterin gets stuck. For it to go to the next cycle, we need to convert dihydrobiopterin back to tetrahydrobiopterin. And this is brought about by the enzyme dihydrobiopterin reductase. Now, this is very important. Again, we'll simplify it. Phenylalanine getting converted to tyrosine, we require the enzyme phenylalanine hydroxylase. It also requires tetrahydrobiopterin, which gets converted to dihydrobiopterin. Then dihydrobiopterin has to be converted back to tetrahydrobiopterin for the next cycle. And in order to achieve that, we require an enzyme called dihydrobiopterin reductase. Now, I mentioned this 2 times for a very specific reason that we'll be discussing about a case phenylketonuria, and we say what is the enzyme deficiency in phenylketonuria? Everybody writes phenylalanine hydroxylase. At MBBS level, this is the bare minimum that you should know. And this does not get you any marks. The specific topic that we are looking for is, are you aware of this dihydrobiopterin reductase also forming a part? Because even if this enzyme is deficient, the patient will present with features of phenylketonuria. This is something which you should understand. Phenylalanine hydroxylase deficiency presents as phenylketonuria. Very good. That is some basic thing which I expect all of you to have known. What we are testing you is, are you aware that there is another enzyme called dihydrobiopterin reductase also involved in this metabolism? Phenylketonuria can be caused by deficiencies of 2 enzymes. There's type 1, type 2, type 3, which is the classical phenylketonuria caused by phenylalanine hydroxylase deficiency. And there's a 4 and a 5, which is involving dihydrobiopterin reductase. So please ensure that this is also written. Next, if this enzyme phenylalanine hydroxylase is deficient, then phenylalanine gets accumulated. Phenylalanine gets converted to phenyl pyruvate. Phenyl pyruvate can be converted to phenyl lactate and phenyl acetate. If you remember the first slide I said, alanine can convert to pyruvate, pyruvate can form lactate, pyruvate can form acetate. The same thing is happening with phenylalanine also. Phenyl pyruvate, phenyl lactate, phenyl acetate. These 3 substances are keto acids and they produce acidosis. So child with phenylketonuria will present with acidosis because phenylalanine is getting converted to phenyl pyruvate, phenyl lactate, and phenyl acetate, all of them which are acidic in nature. Correct? Another thing which we should, so the classical child of phenylketonuria will present with pale skin and hair. There will be mental retardation. There will be mousy odor. And the child will have features of severe acidosis. How does the child present? The child presents with features of severe acidosis. The child has a pale skin and hair. There is evidence of mental retardation. And the telltale mark is that the mother says that the urine of the child or the diaper of the child has a very peculiar smell, which has been attributed to akin to that of a dead rat. Right. I have not smelt it. I don't know it, but the smell is definitely weird. Okay. Now, why does the child present with features of acidosis? As we said, phenylalanine gets converted to phenyl pyruvate, phenyl lactate, phenyl acetate. All of these are acids. This causes acidosis because the enzyme is blocked. The child presents with pale skin. The reason for it is phenylalanine forms tyrosine. Tyrosine is used for the formation of melanin. So when this conversion doesn't occur, melanin formation is decreased, and that results in the pale skin. Mental retardation, multiple theories have been held forward. Phenylalanine is directly toxic to the brain. The byproducts of phenyl metabolism, that is phenyl lactate, phenyl pyruvate, phenyl acetate, they're also toxic to the brain. Tyrosine is required for the production of neurotransmitters, and when they're not produced, that also predisposes to the development of mental retardation. The two major points that we need to know is phenylalanine per se is toxic, and the byproducts of phenylalanine is also toxic. The last question comes, why does the child have a peculiar odor or the mousy odor? The byproducts that accumulate, that is phenyl lactate, phenyl pyruvate, they can produce this distinct smell. They also combine with amino acid glutamine to form phenylactyl glutamine. These are the chemicals that result in the formation of the mousy odor. What we're interested in knowing is phenylacetate, phenylactate, and phenylactyl glutamine. That is phenylactate plus glutamine is phenylactyl glutamine. They're the ones that cause the mousy odor, right? So when we give you the case history, we'll say that a 4-year-old child was brought, a 8-month-old child was brought with features of severe acidosis, mental retardation, pale skin and hair. Mother noticed a very mousy odor. And the question that will link up with it is, why does the child have acidosis? Why does the child have pale hair? Why does the child have mental retardation? What is the biochemical basis of the mousy odor? Right. So is it clear to everybody? Would somebody unmute themselves and explain to me the features why the child has got acidosis? Anyone?
So, on accumulation of phenylalanine, it takes an alternate pathway and forms phenyl pyruvate, phenyl acetate, and phenyl lactate, which are all keto acids. So it leads to acidosis.
Wonderful. Excellent. Somebody else take the question. Why is the child having a pale skin and hair?
When phenylalanine is converted into tyrosine, this does not happen in the case of phenylketonuria. So melanin is not produced. That's why the child has pale.
Excellent. One of the products formed from tyrosine is melanin, and when it's not being produced, it causes the pale thing. What are the causes of mental retardation? Anybody?
So phenylalanine is toxic. So its byproducts are also toxic, which causes mental retardation.
Excellent. Very good. And the last question is, what is the reason for the mousy odor in the child?
Phenylacetate.
Phenyl acetate, phenyl lactate, and phenyl acetyl glutamine. Phenyl lactyl glutamine, right? These products, phenyl acetate, phenyl lactate, combine with glutamine to form phenyl lactyl glutamine, and that is the one which is causing the mousy odor, right? Very good. Glutamine should figure out somewhere in your answer, right? Okay. Now, this is the part of the biochemistry which I'm actually very passionate about, because I see biochemists as the people who would be diagnosing inborn errors of metabolism. The question comes, how do you diagnose a case of PKU? And the answer to that is the classical presentation, a mental retardation, acidosis, mousy odor. Okay. The first simple test you can do is a urine ferric chloride test. Take the urine, add a few drops of ferric chloride. There'll be a transient change in color. Okay, it becomes greenish in color, right? The next thing is urine and serum chromatography. Okay, you must have done chromatography in your 11th and 12th using ink drops and all that stuff, right? Have you done it? Anybody done chromatography using ink and trying to separate the different colors?
Yes, sir.
We have a technique where we can use serum or urine of the patient and do a chromatography, and you will get these kind of bands, each of them pertaining to a particular amino acid. And by that, we can know which was the amino acid in excess in the urine. This is amino acid urea. So if the amino acids are increased, they will come in the urine, and that's called amino acid urea, correct? This is a very crude technique that we can actually do it in our labs also, and we'll show it to you as a demonstration. But a more scientific, quantitative method is what is called the LCMS, liquid chromatography mass spectrometry. If you remember LCMS, very good. It is based on the same chromatography principle that we are doing over here, but the results are more accurate, more sensitive, and we can quantify it. So that makes it a new era in diagnosis, right? So how are we diagnosing a case of PKU? First, we have a clinical suspicion based on mental retardation, acidosis, and the characteristic smell. Following that, we do a ferric chloride test. Take the urine, 1 drop of ferric chloride. You will see that the colors changes to green. A simple test that can be done in any lab is a serum or a urine chromatography, where you can see that each of the amino acids gives separate distinct bands, and you can identify it. This is more of a qualitative. A more sophisticated version is liquid chromatography mass spectrometry, LCMS, where you can get exact identification and quantification of the amino acids in concern. So this is what the world over we are doing for screening amino acid ureas. The next thing which all of you have been talking about is something called the Guthrie's test. And I'll just show you, explain to you what exactly is Guthrie's test. So what happens is that bacteria, when you grow them in a culture medium, they can easily grow. But if you put some chemical in the medium which destroys all the phenylalanine, then the bacteria need phenylalanine to grow. So in this medium where the bacteria was growing, if I grow this bacteria in a medium which does not have phenylalanine, it doesn't grow. Then what I do is I put 1 drop of urine or 1 drop of blood of the patient in the culture plate. This will provide the phenylalanine and the bacteria will start to grow. So if I find that the bacteria are growing here, I infer that the urine has got phenylalanine. Urine will have phenylalanine when the child has got phenylketonuria. This is the Guthrie's test. Let me explain it once again. If you understand the concept, then you can start talking about it, writing about it better. When you put bacteria in a culture plate, the bacteria will all grow. If I make a special culture plate which does not have phenylalanine is very essential for the bacteria. They will not be able to grow, right? Then I put a drop of urine of the particular patient in that culture plate. All over the plate, the bacteria will not grow, but the urine, if it has got phenylalanine, it will provide the required phenylalanine for the bacteria will start to grow. So if the bacteria are growing, like you can see over here, all these areas are empty, but around this disc, you can see the growth of bacteria. So if the bacteria grows, I understand that the urine had phenylalanine, which means that the patient was suffering from phenylketonuria. This was the Guthrie's test. This got improved by the LCMS, where we could do quantitative analysis of the amino acid content in the urine. Right. Nowadays, what we are doing is what's called the dried blood spot collection. You rub the foot of the child nice and proper so that the child is, the foot is pink in color. Make a prick and collect these blood spots. 1, 2, 3, 4. Collect the blood spots in this disc. This is called dried blood spot collection. This can then be couriered to the referral and genetic centers for further testing. What do? We know that the enzyme phenylalanine hydroxylase or which other enzyme? Hello? In phenylketonuria, phenylalanine hydroxylase could be deficient, or there's another enzyme that could be deficient. What is that enzyme?
Dihydrobiopterin reductase.
Excellent. So one of these two enzymes could be there. So we can actually take these, take the enzymes from this dried blood spot and see what is the activity of the enzyme. Is the enzyme working or not? If the enzyme is producing substrate, it'll give me a color, so I know that the enzyme is working. If I don't get a color, I know that the enzyme is deficient. So this is a functional enzyme assay. Nowadays, we can go to molecular and genetic study and pick out the exact mutation which was causing the disease and then label the patient, right? So how do you diagnose a case of phenylketonuria? First is the clinical presentation, then is the urine ferric chloride test, then is a urine or serum chromatography. A more sophisticated version of urine or serum chromatography is liquid chromatography mass spectrometry. We have the Guthrie's test, where the bacteria will not grow in the medium. If you put a drop of urine, the urine provides a phenylalanine, then the bacteria grows. So if the bacteria grows, we know that phenylalanine is present in the urine, which means the child is suffering from phenylketonuria. Then collect dried blood spots, send these samples for functional enzyme assay and do molecular or genetic testing to confirm the diagnosis. So far, so good. Any questions from anyone?
Sir,
Yeah.
is the Guthrie's test specific for phenylalanine, or is it just a concept and we can use it to diagnose other diseases as well?
Very good. Can you build up on it?
As in, if we use another type of bacteria which requires some other chemical or some other compound for its survival, and we use that
Good.
bacteria.
Wonderful. Super logic. You're going one step ahead of time. Really appreciate that. See, what happens is this medium, I have put something which has destroyed the phenylalanine, so the phenylalanine is not available. Correct? So if I put some other chemical which destroys some other amino acid, let's say an alanine or arginine, then I can pick up that amino acid deficiency. So Guthrie's test is basically a concept. Okay, you inhibit an enzyme, make the bacteria dependent, like without this, you inhibit an amino acid. Without this amino acid, the bacteria can't grow. And then you put the urine and see whether the urine is providing that bacteria. It is a concept. Just to prick the bubble for all of you, these were some tests in the 1970s and 80s with LCM, liquid chromatography, mass spectrometry, next-gen sequencing, functional enzyme assay. This has become history. Nobody does it. Okay, much as it sounds very interesting and intriguing, and a lot of our examiners will ask you this question, practically it is not done. But I really appreciate the fact that you are trying to extrapolate the logic to pick up the other amino acids. Wonderful. Great. Good. So now we go ahead with the story. Phenylalanine forms tyrosine. There's a standard question that comes. Tyrosine is not an essential amino acid, but when phenylketonuria, it becomes an essential amino acid. Why? Any takers?
So because phenylalanine forms tyrosine, so
Yeah, phenylalanine forms tyrosine, so tyrosine is not essential. But when phenylalanine can't form tyrosine, tyrosine becomes essential. It's a pretty simple, straightforward question. It'll come as a 1 marker or a 2 marker, so you can use that. Now, tyrosine forms very important products. It could be glucogenic, or it is a glucogenic and ketogenic amino acid. What does it mean? I'll tell it to you in the next slide. It can form important neurotransmitters, dopamine, noradrenaline, and adrenaline. Then we had discussed that the child was having pale skin because of formation of melanin. So tyrosine forms melanin, and if melanin is not there, the child will have pale features. Albinism is because of mutations in melanin production. And the other important thing which we are nowadays discussing with you is formation of thyroid hormone, right? Now, this is the conversion of phenylalanine to fumaric acid and acetoacetate. Phenylalanine to tyrosine, tyrosine to 4-hydroxyphenylpyruvate, that is DOPA, homogentic acid, methyl acetoacetate, fumarate acetoacetate. This breaks down to fumarate and acetoacetate. Acetoacetate is ketogenic. It ends up in forming two molecules of acetyl coenzyme A. Acetyl coenzyme A is ketogenic. Fumarate can enter into the TCA cycle, can go into gluconeogenesis. So phenylalanine can contribute to gluconeogenesis because it ends up in the formation of fumarate. It can also contribute to ketogenesis because it ends up in the formation of acetoacetate, right? So this is something which you should know, that phenylalanine in further steps ends up forming fumarate and acetoacetate. More importantly is this step, homogentic acid being converted to methyl acetoacetic acid. This is brought about by an enzyme called homogentic acid oxidase. Conversion of homogentic acid to methyl acetoacetate is brought about by an enzyme called homogentic acid oxidase. Deficiency of this enzyme leads to a condition called
So alkaptonuria.
alkaptonuria. Wonderful. When this step is blocked, it leads to a condition called alkaptonuria. Now, classically, how the patient with alkaptonuria presents is that you will find darkish discolorations of his skin, of his eyes, sclera. You can, if you look at it properly on the eyes, this part, it's blackish in color. The urine on standing tends to become dark in color. And if you do the Benedict's test, normally this is the Benedict's reagent, a positive reaction will be a red precipitate. Here, the urine turns absolutely black. This is the diagnostic. This is one of the diagnostic tests for alkaptonuria. It's a very simple Benedict's test. You all have done it while doing the urine analysis. This can be used as a screening test to pick up alkaptonuria. Now, what happens is tyrosine to phenylpyruvate to form homogentic acid. Homogentic acid will normally form methyl acetoacetate. But when this enzyme homogentic oxidase is absent, this homogentic acid forms benzoquinone, which eventually polymerizes to form ochronotic pigments. This benzoquinone polymers deposit on the cartilage on the skin. They are black in color. They deposit in the cartilage skin to produce the black color. So this black color that we saw is because of polymers of benzoquinone. They also get excreted in the urine and then get oxidized to form the black pigments. When we do the benzene test, we catalyze that and that gives you the dark pigment. This is alkaptonuria, right? Any doubts about alkaptonuria? This can come as a clinical case for you. Classically, there are patient presenting with pain in the joints and blackish discoloration of his hands and eyes, and urine becoming dark in color on standing. When the Benedict's test was done, it turned black in color. What is the diagnosis? Alkaptonuria. What is the enzyme deficiency? Homogentic acid oxidase.
Sir,
Why? Yeah.
sir, is ochronotic pigment and alkapton body same?
Yeah.
Okay, thank you, sir.
Benzoquinone polymers, ochronotic pigments, they get deposited on the cartilages, they get deposited in the skin.
Thank you, sir.
Okay. So we are good with this. So far, so good. Any questions about this thing? We finished 2 clinical cases as of now. Any questions that anybody wants to ask me?
So, in PKU, you said that phenylacetic acid के साथ और भी 2 components हैं that are giving mousy body odor, but book में तो बस phenylacetate acetic acid को ही mention किया है.
Let's keep things simple. Phenylactate, phenylacetate, phenylpyruvate. Alanine can form lactate, acetate, pyruvate. Just mention that. Also mention that they combine with glutamine to produce a compound which gives the smell. Okay, some books say all the 3, some books say 1. It's better to err on the higher side. You don't lose anything. You just say 1 and say, what about the others? They also produce, right? It's better to err on the higher side than on the lower side.
Okay, sir. Thank you.
Right?
Yes, sir.
Okay, I go to the next one. Okay. The next one is Phenylalanine. Now, have a look at this molecular structure. The point is, nobody is going to ask you to draw molecular structures. But if you look at the molecular structures, you will understand the reaction better. This is phenylalanine. Phenylalanine hydroxylase forms tyrosine. An OH group is being added. Then there's a tyrosine hydroxylase. 2 OH groups get added. Then there is a decarboxylase reaction. What is decarboxylase? CO2 comes out. When the CO2 comes out, what we get is dopamine, right? Phenylalanine, 1 OH group tyrosine, 2 OH group dopa. Then you lose the carbon dioxide, it forms dopamine, right? Then you have a dopamine beta hydroxylase. So now we are adding one more. This is a hydroxylase. We are adding a one more OH group. So this OH group is coming here. So there's a 1, there's a 2, and a 3 OH group. This is norepinephrine. Okay. Norepinephrine or noradrenaline is about the same thing. The British call it one and the Americans call it the other. This, then you add a, this is a NH2. You remove one of the H and add a methyl. Okay. This forms a methyl version of norepinephrine, that is epinephrine or adrenaline, right? This methyl group donation is produced by S-adenosyl methionine. Methionine is an amino acid. Methionine gets charged to form S-adenosyl methionine. S-adenosyl methionine then goes on to donate methyl groups. One of the things to which it donates a methyl group is noradrenaline to adrenaline. Now, what is nor about noradrenaline? In organic chemistry, any group which is being, any methyl group or ethyl group which is being added is donated by the letter R. So norepinephrine, no R. And when the methyl group is added, that is epinephrine, right? So there was something without the R group, norepinephrine. When the R group or the methyl group was added, it became epinephrine. Very good. Is that clear? So far, so good?
Yes, sir.
Okay.
Yes.
The important thing is, one of the questions will be discuss the steps of the formation of the catecholamines. All the epinephrine, norepinephrine are called catecholamines. Describe the step of formation of noradrenaline. This is the step reaction that you should be able to give. Importantly is a condition called Parkinson's disease. Parkinson's disease is a condition when dopamine production in the brain decreases. It's generally being seen quite common nowadays. Incidentally, my mother is also suffering from Parkinson's disease, and it's a very neurodebilitating disorder, right? That is because of dopamine deficiency. There is a very, very rare condition called pheochromocytoma. There's a very rare condition called pheochromocytoma. This is a tumor of the adrenal medulla, which ends up producing excess amount of adrenaline and causes symptoms accordingly. Flushing, high blood pressure, tachycardia, all what you can ascribe to increased adrenaline levels in the blood is caused because of pheochromocytoma. So these will be just about few conditions that can be asked. Right? Any questions on this? Do I go to the next topic? Anyone? Any question? Hello?
No, sir.
Okay, somebody give me some signal so I can proceed further. Now, this is something that we are bashing you up quite a bit to, nowadays with the thyroid function. The tyrosine ends up forming T3 and T4. Has the histology of the thyroid gland been covered?
Yes,
sir.
Okay, so you can help me identify what are these cells? What are these cells?
Follicular cells.
Follicular cells. And what is inside? What is this homogeneous
Colloid.
amorphous material?
Colloid.
Colloid. Excellent. So this is a schematic diagram of the colloid of the thyroid gland, the blood vessel, the thyroid follicle, and the follicular epithelial cells. This is a cross section which is magnified. The iodide enters inside the, one second. The iodine enters into the, is there a marker? Okay. The iodide enters into the cell, passes through the cell. There's this TPO which oxidizes it to produce normal oxygen, normal iodine. Iodine in the traveling in the blood in the form of iodide. It enters into the cell through the follicular epithelial cell, comes to the lumen where the thyroid peroxidase enzyme is present. This converts iodide to free iodine. Step 1. Step 2, the follicular epithelial cells produce thyroglobulins. Thyroglobulins are rich in tyrosine. They get produced and they get secreted into the colloid, right? In the colloid, in the thyroglobulin molecule, the iodine binds to thyroxine to form monoiodothyronine and diiodothyronine. These can together combine to form T3 and T4, right? First, you get iodine inside, then you get the thyroglobulin, which has got tyrosine. The iodine then binds to the tyrosine residues to form monoiodothyronine and diiodothyronine. These can combine. Monoiodo with di will form T3. 2 diiodothyronine will form T4. This is all present in the thyroglobulin. Then the thyroglobulin gets pinocytosed within the follicular cells. The follicular cells first produce the thyroglobulin, which came into the colloid, and after it got iodinated, it once again got pinocytosed and came into the cell, where the thyroglobulin is digested and the T3 and the T4 are made free and then are released on the stimulus. Again, let's say this. Iodide enters into the blood, comes from the blood, enters into the follicular cells, comes to the colloidal side. There is this enzyme thyroperoxidase. It converts it into free iodine and that comes into the colloid. Step 1. Step 2, the follicular cells produce thyroglobulins rich in tyrosine and they produce it into the colloid. Then in the colloid, the iodine is present, the tyrosine residues are present, so they get iodized to form monoiodothyronine and diiodothyronine. These can combine with each other to form T3. That is, MIT plus DIT forms T3, DIT plus DIT forms T4. This is still in the colloid. This is still bound to the thyroglobulin. Then the follicular epithelial cell phagocytizes it, pinocytizes it, and brings it into the cytoplasm of the follicular epithelial cells, where they get digested to produce free T3 and produce free T3 and free T4. Based on the proper stimulus, it gets released out. Right? Any doubts so far?
No, sir.
Anybody doubts? Anybody wants me to repeat it again?
No, sir.
Okay. Anyway, we'll bash this up quite a bit in the practicals when you come tomorrow also. Okay. The thyroid profile, I would kind of try to explain it with an example of the speedometer of a car and driving a car. Now, let's assume that we are having a car and for some reason the brakes are not working. Okay. Very good. So now let's look at it with the car speed and try to make some inferences. Since you don't have the brake, whatever the speed of the car is going to be, you're going to manipulate it with the accelerator, right? So let's assume the car speed is going normal. So the acceleration is normal. Okay, for accelerator, we can think it as TSH also. Now, what happens if the car starts going slow? You're driving the car, suddenly you find the car has started to go slow. So what would be your response? Anybody? You're driving a car, you want to maintain a speed of 40, but suddenly the car started going at 30, 20. What do?
Press the accelerator.
Press the accelerator. You press the accelerator and get the car to come to the normal speed, correct? Okay. Now, what happens if the car, instead of going at 30 to 40, suddenly starts going at 60, 70? You don't have a brake. So what is the next thing that you can do? Next best thing you can do?
Release the accelerator.
Release the accelerator. Release the accelerator. Don't put your foot on the accelerator. Try to get the speed down, right? Now, that's exactly what's happening in the body. What we talk about accelerator in the body, we are talking about TSH, right? If the thyroid levels are low in the body, then the TSH levels will increase, trying to stimulate the thyroid gland to produce more and more of T3 and T4, right? So this is hypothyroidism. In hypothyroidism, what you'll have is the T3 and the T4 are low, but the TSH is high. T3, T4 are low is the speed of the car is low. And TSH is high is you're pressing the accelerator, you're trying to increase the speed of the car, correct? So classical hypothyroidism picture will be a low T3, a low T4, and a high TSH. Does that make sense?
Yes.
Yes.
I sincerely would request you not to go into pattern recognition, but ask yourself questions and build with logic. If you got the logic, you don't have to cram, right? So we're going to a situation where a person's thyroid T3, T4 levels are becoming low. The body will try to compensate it by increasing the TSH. So classical hypothyroidism is a car is going slow. How do you know the car is going slow? The speed is low. What is the speed? T3, T4. T3, T4 is low. And how are you trying to correct it? By stepping on the gas, stepping on the accelerator. So your TSH will be high. This is a classical hypothyroidism. Let's now work the same case in hyperthyroidism. What's happening in hyperthyroidism? The speed of the car is? In
Increase.
hyperthyroidism, the speed of the car is?
Fast.
It's pretty fast. So what are you going to do to correct it? If the speed of the car is fast means your T3 and T4 levels are high. And what are you doing to prevent it?
Decreasing the TSH.
Decreasing the accelerator, removing your foot from the accelerator. So your TSH level will come down, right? In hyperthyroidism, the T3 and the T4 levels are high, and the body, in order to reduce the speed, has taken its foot out of the accelerator. So you're going to get a low TSH and a high T3, high T4. Is that clear?
Yes, sir.
Yes,
Okay.
sir.
Now let's visualize certain question. If you're understanding what I'm saying, let's visualize a condition where the car is going slow and the accelerator is also low. What is this condition going to be?
Or secondary hypothyroidism.
Secondary hypothyroidism. Very good. Your pituitary is not producing TSH. Okay. And because the TSH is not there, the T3, T4 levels are also low. Right? Wonderful. I love this answer. Pretty good. This is secondary hypothyroidism. The car speed is low because you're not pressing the accelerator. So you'll find low T3, low T4, and the TSH is also low. Okay. Now, what about the other case? The car is going fast and the accelerator is also very fast. What condition does that happen?
Secondary hyperthyroidism.
Secondary hyperthyroidism. Okay. Where the TSH production is increased and that's driving the T3, T4 to the thyroid gland to produce more and more T3 and T4. Okay. Now there is a condition called autonomous thyroid nodule. Okay. There's a condition called autonomous thyroid nodule. What does it mean? That there is a rogue nodule in the thyroid gland which is producing excess of T3 and T4. Right? How will you diagnose this condition? What will be the TSH? There is a rogue nodule in the thyroid which is producing excess of T3 and T4. What do you think will be the TSH level in such a patient?
Low.
Low. There is nothing wrong with the pituitary. The moment the pituitary notices that the T3, T4 is very high, it will do everything to bring down the TSH levels. Okay. So in a rogue nodule, autonomous thyroid nodule, you'll find increased T3, T4, but a low TSH. So the last combination is the speed of the car is normal, but I have to press the accelerator hard to make this car maintain that speed. What is this condition?
Subclinical hypothyroidism.
Subclinical hypothyroidism. Excellent. The patient is not hypothyroid because the TSH levels are high and they're inducing the thyroid gland to produce adequate amount of T3, T4. So the patient is euthyroid. But this is subclinical. He is already hypothyroid because the TSH is high. The high TSH is maintaining the T3, T4. Good. So is this concept clear? Is the analogy with the car, the speed, the accelerator, and then correlating with the condition, did it help you make some sense to it?
Yes, sir.
Okay. So that's that for today. Any other question that you need to ask me?
Sir, could you please explain the subclinical part again?
Okay. Supposing I do a T3, T4 and I find that the T3, T4 levels are normal. Is this euthyroid or not euthyroid?
They
Euthyroid is
are.
normal thyroid.
Yes, it's euthyroid.
Yeah, it is euthyroid. But this T3, T4 is being maintained normal because the TSH is doing high. That
Okay,
means
sir.
I'm pushing this thyroid gland harder so that it can maintain the thyroid. So that is subclinical.
Okay,
Okay.
sir. Got it.
So clinically, it is not hypothyroid because the T3, T4 levels are all right. But the fact that TSH levels are high indicates that there's something failing and the thyroid that you have to step on the accelerator to maintain the normal speed. कार में कुछ तो problem है. Right.
Yes, sir.
Good. Any other questions?
So could you please explain pheocytochronia?
Once again, could I explain what again?
Pheocytochronia.